Hello, everybody. First off, I'd like to thank the ABS team for putting this year's conference together. My name is Brendan Mcewen, and today I'll be sharing some of my research from back when traveling for fieldwork was a thing. Batesian mimicry is one of the coolest anti-predator mechanisms to me, and can sort of be thought of as a form of phenotypic parasitism, where a non-toxic species evolves visual signals close enough to a toxic species to fool predators and gain fitness benefits from generalized avoidance without actually having to invest in chemical defenses. So this here is a popular North American example with the coral snake king snake complex, but Batesian mimicry has been observed all over the world in a variety of taxa. Generalized avoidance is the process behind mimicry's utility and functions through something called the cone of protection, which shows how avoidance works in trait space. So here we have a luminance or brightness axis for a toxic species warning signal, where there's continuous variation from darker coloration to brighter coloration. And so the average trait value for the toxic species will lie in the middle. And as with any natural population, there's variation in both directions. Predator avoidance should encompass this whole spectrum. So this trait space is what is referred to as the cone of protection and is considered safe coloration for the toxic species, as well as the phenotypic kind of landing zone for a mimic species to evolve into. Previous work has found that most Batesian mimics are imperfect in that they don't actually fully replicate the traits of the toxic species. But these imperfect mimics still gain benefits of the generalized avoidance because they still fall within the cone of protection from predators. If we take a trait axis from our generalized avoidance, instead of more bright or less bright or more red or towards more yellow or something like that, we can think of it boiling down to just being more conspicuous, as in a brighter luminance or a color that creates a stronger contrast with the background, or less conspicuous on the other end of that spectrum. With this in mind, it could be possible for an imperfect mimic species to fall within the cone of protection while erring on the side of being less conspicuous along each trait axis. So this brings us to our hypothesis that we call cryptic imperfection, which is that mimics should evolve to approximate their toxic model while remaining less conspicuous. To evaluate this hypothesis, we turn to a poison frog complex. We have the poisonous Ameerega bilinguis and the non-toxic mimic Allobates zaparo. And these species are found in the Ecuadorian Amazon. They're about the same size, live in the same microhabitats, eat the same things, are vulnerable to the same predators. And previous studies have demonstrated predator avoidance of the mimic after learning on the toxic model. So Allobates zaparo's mimicry is effective or functional. I'll just refer to them as poison species and mimic species from here on out. Some background contrast data we've collected through visual modeling, image analysis, tells us a few things about the mimicry imperfection in this complex. First, the spots on our poison frog are prominent and bright, intense yellow, while the spots on the mimic are nowhere near as distinct. And this leads to the mimic frogs having a lower contrast with the background, which aligns with our cryptic imperfection hypothesis. Second, however, the dorsum of the mimic turns out to be a bit brighter red and has a higher contrast with its environment compared to the model. So this goes against our hypothesis that the mimic should have less conspicuous traits. But these background contrast data are simply comparing pixel colors from isolated signal components to a leaf background, and don't tell us how well an active observer might find actual frogs in their environment. So to get at that question, we performed a human detection experiment. Detection experiments involve presenting a target stimulus on some sort of background and then tracking how long it takes a participant to find them. And you can use animals in their natural coloration or manipulated color treatments, depending on the question you're trying to get at. Our experiment looked at two species, our toxic species and our mimic species. And we manipulated aspects of some of the stimuli by applying spots from the poison species, the mimic species, or by removing the spots to create no spot treatments. Then we presented these stimuli on a leaf litter background and timed participants in how quickly they were able to find them, which is our proxy of being well camouflaged versus being highly detectable. And these are the results for the detection experiment. On the y-axis, we have reaction time, where lower values represent quicker reactions by participants or increased detectability. And on the x-axis, we have our dorsal color treatments, and grouped into each of those, we have each of the spot treatments. So blue indicates the model spots, orange indicates the mimic spots, and black for no spots. What we found is that for each spot treatment, the mimic stimuli were found more quickly than the model stimuli. And in other words, when you control for spot color, the dorsal coloration of the mimic drives it to be more detectable than the model. And then further, when we compare the actual natural color configurations between model and mimic, we see that the mimic species is still more detectable. So for a recap of what we've seen today, our hypothesis that mimics should evolve to be less conspicuous than the poison species turns out not to be true in this mimicry complex. And instead, it seems that the mimic's dorsal coloration causes it to be more detectable in its environment than the model. So it could perhaps be some sort of exaggeration of the aposematic warning signal that it's evolving to mimic, but we're not entirely sure what's going on there. I'd like to thank a whole bunch of people who've helped me with this work. My advisor, Jonathan Pruitt, my committee, my lab, and in particular, Dr. Jim Barnett, our collaborator in Ecuador, Justin Yeager, and my colleague, Isaac Kinley, for coding our detection experiments, and all of you for hanging in there with me today. I appreciate your time and attention. Enjoy the rest of the conference.